はるばるオランダからお越しいただきましたスリバン先生からスタートです。よろしくお願いいたします。
So I'm very happy to introduce Dr. Stirban. 皆さん、今日の、メインの講演の一つでもありますスティアバン先生の、AGE に関するレクチャーをこれから聞きたいと思います。スティアバン、please.
Konnichiwa. Thank you very much for this very, very kind introduction. Ladies and gentlemen, it's a great pleasure for me to be here and to be able to talk to you about AGEs and also some of the data that we have generated. Just a few words about me. I'm working as a diabetologist and endocrinologist in Germany. I am heading a department in a hospital, but I'm also working in an outpatient unit. And I am teaching also at the university in Düsseldorf in Germany. And let's proceed from Germany to the USA. One of the American actors once said that the short memories of American voters is what keeps our politicians in office. Now, sometimes we would also like very much that our body forgets our sins. Unfortunately, our body does not forget too much. And I would like to show you the data of a study that many of you know, the so-called DCT study, which was a landmark study that changed our understanding about the therapy of diabetes, especially about type 1 diabetes mellitus. And maybe as you know, for this study, patients were randomized to an intensive diabetes treatment, meaning that they received insulin injections at least 3 times a day. And the other group was randomized to a conventional therapy, meaning insulin injections once or twice a day. And those patients were followed up first for 6.5 years. And as you can see on this image, patients that were randomized to the intensive insulin treatment had a better HbA1c as expected than patients randomized to the conventional treatment. And the difference between the two groups at the end of the study was around 1.9% HbA1c. During this period, less microvascular complication developed in the intensive treated group. And you can see here the results on diabetic polyneuropathy. And on the left, you can see that in red, patients intensively treated had less neuropathy than patients that were conventionally treated. And also the nerve conduction velocity was much better in the group intensively treated with insulin. So at the end of this study, we knew that patients with type 1 diabetes are better treated with an intensive insulin therapy. And that's why all patients were switched afterwards on an intensive therapy. And they were followed up until today. And you can see how within a very short period of time, the HbA1c equalized between the two groups. Very interestingly, those patients that were followed up, those that were at the beginning treated with an intensive insulin therapy showed even after 14 years less clinical neuropathy and also an improved nerve conduction velocity compared to their counterparts that were treated at the beginning in a conventional way. That means that somehow the body remembered that during these 6.5 years, intensively treated patients had a better glycemic control, and they took this profit over years with them. And very recently, some data were published of the same group showing that 30 years after the beginning of the DCT, those patients initially intensively treated had less cardiovascular events than those that were treated in a conventional way at the beginning. That means that this metabolic memory lasts for a very, very long time. So that's how this idea, this concept of metabolic memory developed. And of course, we asked ourselves, what is the substrate of this metabolic memory? First of all, how should this substrate be like? It should be influenced by hyperglycemia, and it also should have a long persistence within the body. And you've heard today a lot about advanced glycation end products, and we think that these AGEs are the perfect substrate that explains this metabolic memory. And indeed, data published by Monnier and co-workers have shown that those patients that were in the intensive treated group showed less collagen glycation than the patients that were conventionally treated. And also, around 200 patients from the DCT study received, at the end of the 6.5 years of intervention, a skin biopsy. And the authors measured the concentration of several AGEs, furosin and carboxymethyl-lysine, in the skin biopsies, and followed up these patients for further 10 years. And what they found out was the fact that these glycation products were an excellent predictor for some of the diabetes complications, namely for retinopathy and nephropathy. And this prediction was still significant after correction for mean HbA1c and in multivariate analysis. You've heard about the French scientist Louis Camille Maillard, the one who gave his name to the reaction that leads to the generation of advanced glycation end products. That's why I will not insist on that. But what I would like to stress out once again is that AGEs come into our body by different ways. They are endogenously generated, like the HbA1c, an Amadori product, but they also are absorbed into the body from food or from smoking. Smoking is also an important source for AGEs. I think I don't have to convince you that AGEs act pathogenetically. You've heard today about the way how AGEs can impair cellular function either by modifying proteins, modifying enzymes, or by binding to specific receptors like the so-called RAGE receptors for AGE, inducing thus intracellular oxidative stress, intracellular inflammation. And that's how one can explain why AGEs are involved in the development of diabetes complication. And I would like to take some time to talk about the different microvascular complication of diabetes and the involvement of AGEs. AGEs play an important role in the development of diabetic retinopathy. They can alter pericytes, they increase VEGF expression, they also significantly contribute to the development of diabetic nephropathy, they alter mesangial cells, they also increase VEGF secretion in these cells type 2, and they also increase the secretion of the MCP1 protein. A special activity have AGEs when it comes to the development of diabetic polyneuropathy. Here, they can alter the function of neurons by accumulating within the neurons, but they also can alter the function of Schwann cells by decreasing cell viability, by decreasing replication, and also by increasing the secretion of pro-inflammatory cytokines. But in the case of diabetic polyneuropathy, AGEs play a supplementary role. And this is a very nice paper published a few years ago by the regretted Dr. Bierhaus from Germany. And she was able to show that increased concentration of methylglyoxal, a precursor of AGEs, depolarizes sensory neurons. It increases, thus, electrical excitability and facilitates firing of nociceptive neurons. In other words, high concentration of methylglyoxal induce neuropathic pain. And this is a very important issue because it opens the door for further interventions that might target the neuropathic pain. Also, a very important issue is the action of AGEs in the development of diabetic angiopathy. And AGEs are known for increasing vascular inflammation. They are known for inducing endothelial dysfunction. And about this last topic, I would like to talk to you a little bit more in detail.